Hello everyone. In my talk, I would be discussing a little bit about parasites that are pathogenic in nature, what sort of glycoconjugates they synthesize, and how mass spec comes handy when studying these glycoconjugates. So let's begin with knowing a little bit about the parasites. Parasites are unicellular eukaryotes and they are microscopic just like bacteria or other microbes. They require different hosts for survival and hence they display an extreme adaptation to their environment. They are pathogenic in nature and hence they are able to evade host immune system. As I have mentioned, they require different hosts. They exhibit different life stages to complete their life cycle. Generally, they infect mammalian host via an insect vector. A classic example of that is the Plasmodium parasite, which causes malaria. Other such example of parasitic diseases are African and American trypanosomiasis, which are caused by trypanosome species, and leishmaniasis caused by leishmania parasite species. I assume you all must have read about the life cycle of Plasmodium, so I will present a slightly different example here, which is the life cycle of Trypanosoma brucei. Now, Trypanosoma brucei has two life stages, and the vector of Trypanosoma brucei is a fly called CC fly. Trypanosoma proliferates in the bloodstream of mammalian host, and therefore they are called as bloodstream form parasites, which are these ones. And the bloodstream form parasites, they exhibit a stage-specific proteome and glycome, which I would be discussing in my next few slides. As the number of bloodstream form parasites increase in the bloodstream, they differentiate into stumpy form, which is this one. And when the CC fly, which is the vector, takes the blood meal on a mammalian host, the parasites enter into the insect and they are called as procyclic form. They proliferate in the midgut of the fly, and this procyclic stage again they express stage-specific proteome and glycome, which are distinct from the bloodstream form. Then the procyclic parasites migrate into the salivary gland of the fly, and then where they prepare themselves for the transmission to a new mammalian host. And that's how one life cycle of the Trypanosoma brucei is completed. So, when we say mammalian pathogens, it does not always mean human pathogens. Many of the parasites infect animals and livestock. For example, Plasmodium falciparum, Leishmania, Trypanosoma brucei gambiense are human pathogens. But Trypanosoma brucei is an animal pathogen. That is why Trypanosoma brucei serves as a model organism to study the human pathogens. My talk will mostly revolve around Trypanosoma brucei, and every time I'm saying Trypanosoma brucei means Trypanosoma brucei, which is not a human pathogen. Now, the most common feature of the parasites is the synthesis of glycoconjugates and glycan binding proteins for protection and to interact and respond to changes in their environment. Especially GPI anchored surface glycoconjugates are highly abundant in parasites, which provide the first line of defense when they are in mammalian host. So here the cartoon represents the surface molecules of Trypanosoma brucei. As I mentioned earlier, that Trypanosoma brucei expresses life stage specific proteins and glycans, and there are two life stages, bloodstream form and procyclic form. The bloodstream form parasites express VSG, which is called variant surface glycoproteins, and the entire surface of the parasites is densely packed with VSGs, and there are 5 million copies of VSG per cell. And similarly, in the procyclic form, they express a protein called procyclin, which is again a surface glycoprotein, and there are like 2.5 million copies per cell of procyclin. Both VSGs and procyclins are GPI anchored as well as N-glycosylated protein. The Trypanosoma brucei parasites, especially the bloodstream form parasites, they have evolved a process called antigenic variation, where they switch the expression to alternate VSG's expression from a large repertoire of a silent gene in order to maintain the infection. The different VSG variants can also differ in the extent of glycosylation. That is how trypanosomes are able to evade host immune system and maintain the infection. So let's discuss what a GPI anchor is and what type of glycoconjugates are being expressed. The term GPI stands for glycosyl phosphatidylinositol, and as the name suggests, it is an anchor, which means it anchors the glycans and the proteins to the membrane. It is one of the biggest and most complex anchor known so far. GPI anchor is made of lipid and glycans. The lipid can be an alkyl acyl glycerol based lipid or a ceramide based lipid. The lipid is then attached to the inositol ring via a phosphate group, and then the inositol is further attached to a glucosamine residues and then to further three mannose residues. Now the proteins are attached to the third mannose residues via their C term and with a phosphoethanolamine group. Now this glycan core or the mannose residues can further be modified with glycan side chains, or there might be some other chemical modifications as well, which basically add on to the complexity of the GPI anchor molecule. For example, you can see the bloodstream form VSG glycoprotein and procyclic glycoprotein. VSGs have a diacyl glycerol lipid backbone and they have a less complicated glycan side chain. VSGs are further modified with N-glycosylation, and the procyclin have a monoacyl glycerol based lipid in the GPI anchor and a very complicated glycan core, and they have the glycan core is mainly modified with galactose and glucanac repeats, which are called polylactic repeats, and they are often capped with alpha-2,3-sialic acid. And procyclins are also N-glycosylated, mainly with oligomannose structures. So we talked about the glycoproteins, but there are glycolipids as well. For example, here I'm showing the Trypanosoma cruzi glycolipids. The surface of the Trypanosoma cruzi is densely covered with mucins and gippals. Gippals are the glycolipids. And the Gippal term stands for glycosyl inositol phospholipids, which are the most abundant surface glycoconjugates of Trypanosoma cruzi. Now, the structure of the gippals is the same as the GPI anchored molecule, and where a lipid is attached to a glycan core, and this glycan core can further be modified with several other glycan side chains or some chemical modifications. Now, the gippals in Trypanosoma cruzi, they play a very important role in establishment and maintenance of the infection. So we have understood so far that the glycosylation is highly abundant in parasites and is very crucial for parasite survival and infection. So now I'm going to talk about some of the methods that we use to study the glycoconjugate molecules, and these methods are mainly mass spec based analysis method. So in my talk, I would cover four different methods. First would be to study the complex and glycans of bloodstream form Trypanosoma brucei, and where we employ GCMS-based strategy. Next would be to analyze the GPI glycans of procyclin, and where we employ the electrospray mass spec. Third would be to study the GPI anchor precursors, the precursors that are important in the biosynthesis pathway, and we would analyze the precursors with electrospray mass spec again. And the fourth one would be to study the gippals, the Trypanosoma cruzi glycolipids. And again, we will study the lipids with electrospray mass spec.